Okay, so when we left off, we talked about the need for different locations for receptors. If the thing freely diffuses across a membrane, then the receptor is going to have to be internal versus if it is something that the membrane is impermeable to, then the receptor is going to have to be on the surface. This is not just going to be the receptor location, but also how we release it. If it's impermeable, if the membrane is impermeable to the signaling molecule, then you can make a bunch of it and load it up into vesicles and have a regulated release. Whereas if it is going to freely diffuse across the membrane, then you're going to need to produce it only when you need to release it. And so that's a big difference between the steroid hormones and the proteinaceous hormones. Another thing that is really important in understanding signaling is that signaling usually happens in a signaling cascade or a signaling pathway. And so there are going to be multiple steps, and you're going to have a signaling molecule that is received by the cell, and then there will be some sort of cascade that it sets off. And there can be lots of downstream effector proteins. Maybe an enzyme gets turned on, or a change in cytoskeletal protein structure, or maybe some transcriptional activity. But it's usually going to be at the downstream side of a signaling pathway. And this is important because this allows for two things: amplification and integration. So remember when we talked about maybe these three signals might say to survive, but add in two more, and it might say divide, and add in two different ones, and it might say differentiate. Well, the only way that we could have that is if we had signal integration from multiple pathways. And so very often the extracellular signal will be received by a primary transduction pathway that will allow for the signal to be received on the cell surface, and then there will be maybe some enzymatic amplification. So if it turns on an enzyme, that enzyme is going to do many iterations of its activity, whatever it is. That would create multiple downstream effectors past that first signal. So in the visual cascade, for instance, one photon is going to turn on one rhodopsin, but then when we transduce that signal, the transducins and so we're activating a G protein coupled receptor, that's going to turn on an enzyme that is going to have many amplifications. And so one photon is going to result in maybe tens of thousands of extra potassiums moving through that membrane because of multiple channels, because of multiple enzymes turning on multiple enzymes. And so this is going to give you an amplification of any signal that you receive. Likewise, there can be multiple pathways acting on a central point, and what this allows for is maybe one signal is not enough to create differentiation, but a signal from one pathway and a signal from another will cause differentiation. Or maybe say like you're getting a death signal from one place but a survive signal from another place. So these signals pathways may be in opposition to each other, and then it's kind of like a teeter-totter. You're going to go in the direction where there's more signal. So these signaling pathways might inhibit each other, and that allows for integration. And so this is an important thing to keep in mind when we talk about cell signaling, is that these signal transduction pathways, they have multiple places where they interact with each other, and this allows for a greater diversity of signaling, just one signaling molecule, because of multiple receptors, multiple cell types, multiple pathways, and then other signaling pathways amplifying or inhibiting any pathway. You can get a vast array of responses to any one signal. This also creates the Gordian knot of trying to figure out how these pathways interact with each other. And this is why there's so much work going on in this area of biology, is because it's not obvious. It's not really easy to tease out what one signaling molecule might be doing in a particular biological effect. So in general, there's two real common ways that we have downstream effects. One is through the phosphorylation. We talked about this way back in chapter 4 when we were talking about how proteins get turned on and off by phosphorylating them. And of course, you guys looked at a protein phosphatase, and the acid phosphatase in lab. And so the phosphorylation of a protein might turn it on, and then it would be maybe an active enzyme. Maybe it's a kinase itself, and by being phosphorylated, now it can phosphorylate other targets. So turning on an enzyme by phosphorylating it is a really common mechanism. Now, I don't want you to think that phosphorylation always turns an enzyme on. Phosphorylation can also turn an enzyme off. This is just an example of turning an enzyme on by phosphorylating it. Likewise, there's another really common mechanism. We'll see this in G protein couple receptors, and that's by GTP binding to something to turn it on, and then the hydrolysis of the GTP to GDP turns it off. And we saw this in import into the nucleus with RAN-GTP GDP system. And we're also going to see this in the cytoskeleton with tubulin. So this is another really important signaling mechanism, the GTP binding. And so those are two common paradigms for signaling. And then, of course, the one that you are probably really familiar with is opening an ion channel. The ligand-gated ion channel is holding maybe sodium on the outside of the cell, and then when the ligand binds to the receptor, the channel opens and sodium rushes in, and that changes the physiology of the cell. And so this doesn't have to be sodium, it could be potassium, it could be calcium, it could be chloride. It doesn't have to be rushing in, it could be rushing out, but these ligand-gated ion channels, or ion channel coupled receptors, are one of the main cell surface receptors. Another very common cell surface receptor is the G protein coupled receptor. We talked about this when we talked about membrane proteins. This is a 7-pass transmembrane protein and the most common gene family in your genome. There's over 1,000 genes that code for G protein coupled receptors. So the signaling molecule binds to the G protein coupled receptor, and it causes some sort of conformational change that allows it to maybe interact with the G protein, maybe release a G protein, and then there's an activity. Usually there's going to be an enzyme downstream of this that will get activated by the G protein. And we'll talk a lot about G protein coupled receptors. The signaling molecule doesn't have to be a signaling molecule at all. Of course, the visual cascade is light hits the G protein coupled receptor and causes the 11-cis retinol to turn to all-trans retinol and changes the conformation that allows it to interact with its G protein. So that's the second major class. The third major class is the receptor tyrosine kinase, or the enzyme-coupled receptors. So here the signaling molecule might bring two of the same kind of receptor together, or it may be heterologous coupling, so two different. So maybe they're the same, maybe they're different, but in either case, when the signaling molecule binds to the receptor, this allows for the activation of that receptor. So very often there's in receptor tyrosine kinases, there's a cross-phosphorylation that may activate the enzyme. It doesn't have to activate an enzyme. Maybe now it just becomes a good scaffolding molecule for an enzyme to bind to, and that activates that enzyme. And so it could be multiple steps to get to an activated enzyme. And we're going to look at some of those in this chapter. We're going to move on to G protein coupled receptors, so move on to the next